In this lecture, we're going to take a closer look at the mechanisms underlying the action potential. And we begin here on the upper left-hand side where we have a ligand-gated ion channel. In this particular case, it's a sodium channel that's also referred to as the nicotinic acetylcholine receptor. And this is the one we find at the junction of the motor neuron axon terminal and a skeletal muscle fiber at the neuromuscular junction. And once we have acetylcholine binding to a single ion channel, sodium channel, at the location where the terminal axon interacts in synapses with the muscle cell, we see it takes two acetylcholine molecules on one sodium channel. And when that sodium channel opens up, it will result in the movement of these sodium ions through the membrane, and the one of the sodium ions will interact with the interior portion of the voltage-gated sodium channel located just distal to the neuromuscular junction. And when that happens, we see that the negative state of the membrane potential on the inside switches over to the positive state, and that was the positive 30 millivolts at the peak of the depolarization. And that results in what's called fast channel opening. And so when that channel opens, it then will allow the further depolarization to take place. And this is what really drives the interior of the cell in this particular region to positive 30 millivolts. And that will initiate a slow closing of the inactivation gates. So the fast opening of the activation gate is triggered by the weak binding of sodium on the interior part of the channel. And that triggers the slow inactivation gate to close, and that will eventually move the channel into its inactivated or closed state. Note that this is not the initial resting state because we still have that positive environment inside the cell. And so when we talk about the depolarized state at approximately plus 30 millivolts on the inside of the cell, we know that the sodium channels are closed, and that closure is generated by this analogy here of a very slow moving gate that will close a channel until it fully restores back to its resting state where the activation gate closes the channel. And when that happens, we have a negative state, a negative potential inside the cell. And we know that the depolarized state will trigger the repolarization generated by the movement of potassium from the inside to the outside of the cell. If we take a look at the two main players here, the first is the sodium channel, and the sodium channel is a voltage-gated single long protein or polypeptide that is organized into four distinct domains. It's important to note that the ligand-gated sodium channel has a very similar structure, but there are some differences, particularly with the ability to form a chemical bond with the neurotransmitter. But in terms of the voltage-gated sodium channel, it's characterized by having 6 transmembrane alpha helices, and that's illustrated in this picture that I'll get to here in a moment. So the voltage change across the membrane is sufficient to cause a slight change in the structure of this particular protein, and so the depolarization is said to twist the protein configuration that will open up the pore. And it's important to note here that when we talk about neurons in a general way, the neuron membranes typically have a relatively small density of sodium channels, which makes up a little bit less than 1% of the total membrane or the neurolemma of neurons, but these small number of sodium channels is still sufficient to allow communication along the axon of these neurons. And this particular channel acting individually is very fast. And so some estimates will place about 10 million sodium ions, and this number does vary a little bit, but approximately through one channel, 10 million sodium ions is thought to transit through in about 1 second. What you're looking at here on the bottom of the slide is one of the first depictions of the sodium channel. This is a hand-drawn image from this paper published in 1995, and it's considered one of the classic papers in terms of revealing the chemical and amino acid sequence of the sodium channel. The next major player in terms of the action potential is what we see after peak depolarization, which is repolarization, and that's generated largely by the potassium channel. So the potassium channel is composed of 4 identical subunits, and that's illustrated on this computer-generated image here at the bottom. This is in a sense, a bird's eye view down the pore, where we see a fairly symmetrical protein made up of these 4 subunits. And when these 4 subunits are brought together, it forms a cone-shaped pore, and that's illustrated on the right-hand side of this image, where you see on the outside of the cell, the channel has a wider dimension as compared to the more narrow dimension on the interior of the cell. And this channel was initially discovered in mutated fruit flies that tend to shake violently when in the presence of anesthesia. And it was determined that these fruit flies that have this mutated channel as a result of what's called the shaker gene, which is a mutated gene that produced a channel that produced unstable action potentials and resulting in unstable muscular contractions in fruit flies when those fruit flies were in the presence of ether. And one last point here is that Roderick McKinnon is the scientist credited with this discovery of the shaker gene, and as a result was able to provide a detailed description of the structure of the potassium channel. We take a closer look at the potassium channel. We start with the outside of the cell with a larger dimension than the interior of the cell, which has a narrower dimension. And so, although we see a cone-shaped appearance from outside to inside for the entire channel, the pore actually constricts from inside towards the outside. Remember, potassium is transiting from the interior of the cell towards the exterior of the cell. And there are 2 major parts to the pore. The first major part is the first 22 angstrom component, has a 10 angstrom diameter, where the potassium ion essentially moves in water, and that's illustrated here by the blue. The remaining portion of this very constricted region is called the selectivity filter. And this is what gives the potassium channel its very unique selectivity in terms of allowing potassium and only potassium through the channel. And so you note here the constriction changes from approximately 10 angstroms on the opening of the channel of the pore to a very narrow 3 angstrom diameter in that selectivity region. And what is key for potassium transit through that selectivity pore is the ability of potassium to lose H2O molecules. So that begs the question of why does potassium and only potassium move through the potassium channel? And it's known that potassium channels are 100-fold more permeable to potassium than sodium. So just to review from slide 40, recall that the filter diameter, the selectivity filter, is estimated to be approximately 3 angstroms in diameter. So the filter can reject ions with a radius greater than 1.5 angstroms, which is half the diameter. So ions that are larger than potassium or larger that can fit through this can easily be rejected by the selectivity filter. However, the problem we have here is if we take a look at the ionic radius of potassium. The ionic radius of potassium is 1.33 angstroms. If we multiply that by 2, we get 2.66 in terms of a diameter, which says that it can fit quite easily through the selectivity filter because it's smaller than the maximum width of that filter. If we take a look at the sodium ion, however, the sodium ionic radius is quite a bit smaller at 0.95 angstroms than potassium. So if we take a look at sodium, its diameter is less than 2 angstroms. So in theory, it should be able to move through the filter, but it does not. It's actually unable to move through the potassium channel. So the question here is sodium can fit in terms of its size, but it doesn't go through. The answer to this question, which will not be provided in this video, can be accessed at this link here. And so what this link is the scientific presentation Professor McKinnon gave at his Nobel Prize ceremony. And in the first part of this presentation, he talks in depth about why potassium can go through and why sodium, despite its smaller size, cannot go through the channel, particularly the selectivity filter. So I'll leave it up to you to link to Roderick McKinnon's Nobel Prize scientific presentation, and that will provide the answer to this question.